All right, these will be your notes on mitosis and the cell cycle. These notes were given on 9/28. So your notes will consist of drawing a circle large enough to fit, I'll give you a preview, this much text into each quadrant. And I'm going to talk through what each of these quadrants is all about. So that's an idea of how much needs to fit in. We'll go through each quadrant a little slower. Okay, first thing you'll need to do is make a space in that upper left-hand quadrant for mitosis and karyokinesis. Karyokinesis is just the technical term for mitosis. So karyokinesis means the movement of DNA, which is what mitosis is. Okay, this whole section here is interphase. Interphase is what the cell goes through, so all of this area here, this will all be interphase. Interphase is what the cell is in 95% of the time. It's when the cell is performing normal cellular functions, just doing its little cell thing. And then we'll talk about what each of these parts of interphase are. This is mitosis, and it accounts for only about 5%. Sorry, that 5% kind of got jumbled in there. It accounts for about 5% of the cell's life. So even though it's 25% of this pie, it's only about 5% of the cell's actual life. Mitosis is broken up into 4 stages: prophase, metaphase, anaphase, and telophase. The end of mitosis results in cytokinesis. Cytokinesis is not a part of mitosis. Cytokinesis just means cyto is cell, kinesis is movement. So when the 2 new cells move apart. Just like karyokinesis means when DNA, karyo, moves apart, kinesis. Mitosis is the division of one cell into 2 cells. So it's the movement of DNA into 2 different cells, DNA movement, karyokinesis, in order to make 2 cells move apart from each other, cytokinesis. The first part of interphase is G1. Remember, at any point in time, you can pause these notes to catch up, slow me down, speed me up, whatever you'd like. So the G1 phase is the gap or growth phase, and that's where RNA and protein is synthesized. It's also where cell growth occurs and where nuclear growth occurs in order to make room for the S phase. The S phase is where DNA is actually duplicated. So in the S phase, or synthesis phase, DNA is synthesized, which just means made. So an existing strand of DNA makes a copy of itself to form 2 complementary strands of DNA. Also in S phase, histones are synthesized. They play a part in mitosis. And centrioles duplicate, also play a part in mitosis. Last is the G2 phase, or the next gap or growth phase. Cell organelles duplicate. Cell growth continues. Nuclear growth continues. And this is the last checkpoint before mitosis begins. So before DNA is, not copied, that's incorrect. Before DNA is separated. DNA is separated in prophase, metaphase, anaphase, and telophase until eventually 2 new cells are created. That's the lab that we'll do.